All right. Well, hi, everybody. I'm Matt Moore. I'm president-elect of Phi Tau Sigma. Thank you for taking the time to join us today. So I'm happy to introduce Dr. Lutz Grossmann, who will be giving us a talk. Um, Lutz did his bachelor's, master's, and PhD at the University of Hohenheim, as well as a postdoc. He's held numerous visiting scientist positions in a number of very prestigious institutions, including University of Guelph, as well as CSIRO in Australia. He's published 65 peer-reviewed publications, has over $3.5 million of grant funding, published 3 books, and has had over 30 invited talks. And so we're really excited to hear about his really, really interesting research, sort of on the future of foods. And so I'll hand it off to Dr. Grossman. So thank you again for being willing to provide a talk about all your cool work.
Yes, thanks, Matt, for the introduction, and welcome to everyone to the seminar. I will go ahead and share my screen. Okay, does it work?
Let's see. Yep, you're good.
Perfect. Yeah, so welcome and good afternoon or good morning. I'm not sure where everyone is. But yeah, my name is Lutz. I'm an assistant professor in the Department of Food Science at the University of Massachusetts. And I want to talk a little bit about the research that we are doing in my lab and the work that my group has been doing over the past few years. And our group really focuses on how can we make ingredients that are more sustainable, but also are really functional. So we're looking at this from a biomanufacturing perspective, but also from a food science, food technology, food functionality perspective, and a food engineering perspective. And I want to talk to you today about 2 really specific projects that we are working on and introduce a little bit more of what we are doing in this research. And most of my research is really focused on sustainable proteins. Yeah, not everyone knows me, so Matt already briefly introduced me, and here's just like a broad overview, just a brief background. I grew up in Germany, and then I studied food science and technology at the University of Hohenheim. And during my master's, I went to CSIRO in Australia to do a research stay, which was really cool. And then during my PhD, I stayed in China at Changnan University and at University of Guelph, which was also both really, really cool experience. And everyone who is currently doing a PhD and is not an international student, I would highly recommend to get some international experience and to see the world while you're still a student. It really opens up a lot of opportunities, and it's a really good learning experience. So grab every experience that you can get. Yeah, and then I finished in 2019 with my PhD, and then started at UMass Amherst now for more than 4 years ago. And when I started UMass, I was really thinking, what kind of research do I want to do? And during my PhD, I worked with microalgae as a novel protein source. And it was really interesting. The research was really interesting, but I also realized that microalgae production itself has a lot of limitations. And then when I started at UMass, I wanted to kind of build on this experience on utilizing and understanding novel ingredients and how we can use them in food matrices. But I wanted to also expand what I've been doing during my PhD. And based on the limitations that I saw with microalgae cultivation and production, I was looking into some other biomanufacturing technologies that could overcome some of the limitations that microalgae cultivation has, especially in terms of that you need these large bioreactors and large surface areas that really limits a little bit its potential. And that's why I started to look more into these novel upstream cultivation methods. And I will introduce one today, which is the hydrogen fermentation. But we are also interested in, for example, microprotein biomanufacturing, so growing fungi in a bioreactor and then trying to understand how we can use the the mycelium, so the fungi, quote unquote, roots in food matrices. And this is kind of under the umbrella of novel upstream cultivation methods. So everything that's related to food biomanufacturing, or broadly speaking, food biotech. But as a food scientist, like the whole food biomanufacturing is interesting, but I'm also interested in what can we then do after the bioreactor. So because you can have like a really nice product or ingredient that comes from the bioreactor, but if you can't really translate it into a food, then it won't be successful. And that's why my lab is also interested in then extracting proteins, characterizing them, so characterizing their properties, especially in terms of, for example, can they foam, can they make emulsions, can they gel? And just understanding the properties of these novel ingredients on a fundamental level, because this then helps us in the final step in really making food structures and trying to translate these ingredients into actual food matrices, and doing so by leveraging technology. So we are especially interested in extrusion, but I could also see myself in the future that we are also looking into other technologies that can be used to really structure these novel ingredients into foods. But currently we do really a lot on extrusion work. The raw materials we're working with is quite broad. So we're working with plant materials, bacteria, fungi, and also a little bit still with algae. So that's kind of the broad overview of my group. And I want to highlight like two research areas that we do, like one on the novel upstream cultivation methods and another one on extrusion. So the part one will be on the novel upstream cultivation methods. And as I already briefly mentioned, we are really interested in finding new ways to make foods. And one way of doing this is to use food biomanufacturing. And imagine you probably are very well aware of protein powders, like think about your whey protein concentrates, or your plant protein concentrate, for example, soy protein concentrate or pea protein concentrate. And those protein powders, when you buy them, they have like a protein content of around 60 to 70%, especially the concentrates. But those are to get to this high protein content, you have to do a lot of processing. So you have to isolate the proteins, you have to precipitate them, you potentially have to run them over a membrane filtration unit, you have to separate a lot of other fractions, a lot of other ingredients that are naturally present in the raw materials, to get to this really 60 to 70% of protein. But imagine you can have a technology that provides you just straight from the production a 60% true protein content without doing any refinement. So without doing any separation, any precipitation, like just you do your production, your raw material production, and delivers you 60% of protein. And there are not too many technologies out there that can do this, but food biomanufacturing, so making ingredients in foods in a bioreactor, can easily do this. And the question would be, why are we actually interested in this? So why do we want to make foods in a bioreactor? And I'm talking more about, not too much about cultivated meat, which is also sometimes referred to making food in a bioreactor. I'm talking more about like growing, for example, bacteria or fungi in a bioreactor to make foods. So what is actually the motivation behind this, and why is it a good idea to research and a really interesting research field? So here you can see the overall agricultural production globally. And what you can see is that only 15 crops provide 73% of agricultural products. So there's actually not a huge variety in crops that basically feed the world. Not all of this, what you can see here, this is just like the FAO statistics. Not all of this goes to food, like especially sugar cane and corn. Some of this also goes to ethanol production. But overall, there's not a huge variety in crops that feed the world. So it would be maybe a good idea to also establish some other production methods that increase the variety and make the food system more resilient. And it gets even more interesting and more limited when you look into where does actually the protein of our diet comes from. And this is then really specific for food. And here you can see the numbers for the USA. So in the US, only 4 plants and animals provide 75% of the protein that is consumed in the US. And this is wheat, chicken, cows, and pigs. So those 4 plants and animals provide 70% of the consumed protein in the US. So there's not a huge variety actually when you look into the data. And this causes some issues with supply chain issues. If there's like a disease that comes up in the supply chain, can have huge ripple effects. And so it's beneficial to think about what other production methods are out there that could provide protein in the food system. And one way to think about this in general on how we make foods. So in general, the majority of foods comes from agricultural production. But then is this actually the only way we can make foods? We are kind of really accustomed to it because we do it since thousands of years. But when you just think about it, maybe we can also think about other ways to make foods that do not necessarily rely on agricultural production, especially when we look at the land area we need for agricultural production and some other negative environmental effects. I'm not saying that agricultural production is something that should be abolished, but I'm saying maybe we can think about other ways to make foods that could supplement agricultural production. And this way, maybe lower the amount of land we use for making foods. And this already happened before, where we changed from one production type to make foods, in this type, in this case, ice, to another way, basically using electricity to make ice. And the story behind this is like this guy here. His name is Frederick Tudor, and he was also called the Ice King of Boston. And he used to be really, really rich. So at the times, he was one of the richest persons in the US. And his business was basically to sell ice from the ponds of Massachusetts. So Massachusetts has a lot of ponds and small lakes, and they all froze over in the winter. And what he did, he harvested the ice from these ponds and lakes, and then shipped it all around the world and across the US and sell it. So what he basically used was the land area of Massachusetts ponds and lakes, and sold the product from this land area all over the world. And so nowadays you would think, oh, that's really, really inefficient. Why would you, like, first of all, wait until all of these lakes freeze over, and then you have to get it from the lakes, and then you have to put it onto ships until it reaches like the Caribbean, for example, half of the ice is gone. So nowadays you would say it's not a really efficient way to make ice. And that's why, especially after after the First World War, when electric fridges became really, really common, this whole business model basically collapsed, because it was much more efficient to use electricity to make ice and then have it available at any time, basically, in your home. And so this completely switched on how we made ice. Previously, we made ice from basically waiting until the winter comes and then try to also store it over the summer. But nowadays, we make ice from electricity in our fridge. And this also saved us a lot of land use. So we don't need lakes and ponds anymore to make ice, but we can just make it really, really efficiently in a really small area in our homes. And maybe we can apply the same to agricultural production. So is there a way that we could use electricity to make food ingredients that would require much less land and maybe also fewer natural resources? So the same idea, but maybe we can apply the same idea also to other production methods. And this is in general possible. It's also not necessarily a completely new idea, especially in the 60s when space exploration became more available and doable. Like people were thinking, how could we feed astronauts on really long-term space missions? And then they were thinking about using electricity to make foods in a way. And they already figured out that you can make hydrogen through electricity and then feed hydrogen to organisms that then use the hydrogen to make their to multiply and then make edible biomass. So this was kind of idea that already was around in the 60s, but it was never really scaled for a reason that was mainly related to electricity costs and especially the costs of renewable electricity. But because electricity up to a few years or decades ago was mainly produced from fossil fuels and nuclear power, so technologies that are not necessarily the cleanest. But now we have like really, really cheap renewable electricity. So renewable electricity, as you can see here, especially wind and solar became like really, really price competitive and now is regularly cheaper than any other sources of electricity. And we can use this electricity to make green hydrogen. So hydrogen from renewable electricity. And this allows us to produce food ingredients through hydrogen fermentation in a really in a way that is based on renewable energy and has a low environmental impact. And this is only possible since a few years basically. And that's why we are really, really interested in this new technology of hydrogen fermentation. So how does it actually work? So now I talked a little bit about it. So how does it work and where does the idea come from? So the technology is based on bacteria that are called hydrogen oxidizing bacteria or hydrogenotrophs. So you can find them almost in many, many areas around the world. One common example is you have these deep sea vents, and these deep sea vents, they release a lot of gases, including hydrogen. And you can find hydrogen oxidizing bacteria really close to these deep sea vents, but you don't necessarily have to go to the really deep ocean to find these bacteria. The organism that we are using was actually found or isolated just in a river sludge in Germany. So everywhere where there is anaerobic fermentation, usually those anaerobic bacteria, they also always release hydrogen gas. And then there's usually a symbiosis going on with hydrogen oxidizing bacteria. And that's what's happening often in these river sludges. And that's where the organism we are using was isolated, which is called Cupravidus necator. So those organisms, they use hydrogen, they oxidize hydrogen, and this oxidation process releases energy that they can use for their own metabolism. And what they use is the Knall gas reaction. So they use hydrogen and oxygen and basically carry out the Knall gas reaction in their cells. There are also other chemoautotrophic bacteria, for example, sulfur bacteria, iron bacteria, nitrate bacteria, that kind of use a similar mechanism of using non-organic compounds to produce energy. But the nice thing about hydrogen is really that we can produce it from water. So we can use electricity to split water and then make hydrogen, and then the bacteria do the reverse reaction and basically regain the energy. And yeah, this is the reaction that they are doing in the cell. So in this case here, you can see you have a hydrogen and oxygen atmosphere. And if you give it a little bit of of activation energy, it releases all this energy. Here you can see it burns, so it releases all the energy as heat. But the organism figured out a way to really harvest this energy not as heat, but as to store this energy as chemical energy, or to transform the energy into chemical energy and then basically make biomass. And this is exactly the reaction what you can see here. So this reaction releases around 286 kilojoule per mole. And as food scientists, we like to talk in kilocalories. So here you can see like 1 liter of hydrogen. I forgot what pressure conditions this is for. But 1 liter of hydrogen under pressure has around 2.6 kilocalories. So this is just energy that a bacteria or this organism can use. And then it uses this for cell growth, and in the end, biomass that we can then harvest in a bioreactor. And here you can see how the cell is doing it. It's basically a mix of the dark reaction, of the light independent reaction, of the photosynthesis metabolism of plants. And this organism is also really, really old. So this is really a metabolic pathway that is like thousands or probably millions of years old. And you can see it's a Calvin cycle where the cell fixes CO2. It then has a few enzymes. So you can see here we provide the cell hydrogen. It then takes up the hydrogen into the cell, oxidizes the hydrogen with an enzyme that's called hydrogenase. It releases the electrons, and instead of that, all of this just goes to heat. It then harvests the energy in the electron transport chain, makes water in the end, makes like the typical your proton gradient, uses this energy to make ATP, and then uses this in the Calvin cycle to fix CO2, and then makes all of these sugar components from this energy that's being released from the hydrogen oxidation. And this in the end results in biomass. So it's really a kind of, I think, really interesting metabolic pathway. And that's why I like microbiology. You would never think that something like this exists, but it's so crazy what different kind of metabolic pathways always exist, where I think, huh, how is this even possible? But yeah, it's really, really interesting. And we can use this really to drive food biomanufacturing. And here you can see how we kind of envision this. So the whole process is based on renewable energy. So we make electricity using solar, wind, or geothermal or hydropower. We use this electricity to drive an electrolyzer. So this splits water into hydrogen and oxygen. We can fix CO2 from the atmosphere, or you can use a CO2 off gas from an industrial production or from a heterotrophic fermentation process. To think about, for example, you have a brewery that releases oxygen during CO2 during the ethanol fermentation. And you could use this CO2 then in this heterotrophic and this autotrophic fermentation process. So any CO2 off gas you could use here. And then obviously you also have to feed it some micronutrients. So you have to feed it a nitrogen source, ammonia, phosphates, and minerals. You feed all of this into a bioreactor. So you bubble the gases in, and you have a liquid media that is rich in the micronutrients. You bubble all the gases in. You wait a few days, then you get a protein-rich single-cell biomass out of it. You harvest the proteins, the biomass, and this is then a single-cell protein. So this is kind of how we envision the overall process. And the question is this actually really an efficient way to make foods? And here's what you would expect in terms of yields for different types of production methods. So you see here on the right, it's like your typical soybean field, which is one of the most efficient crops in terms of protein production. And if you have like 1 hectare, or which is around 2.5 acres of a soybean field, it provides around 1.1 tons of protein per year. And with this, you can feed around 30 people, depending on how much protein they consume. But on average, it's like around 40 people. If you do a heterotrophic production, so think about your typical mycoprotein production, so a mycelium production, there you need to make sugars for the production, for the biomanufacturing. So you need to have a sugar beet field or a corn field that you then use to make fermentable sugars. And with this process, it's already a little bit more efficient. You can make around 2.7 tons of protein per year, and you can feed around 90 people. Then if you look at the land efficiency of the hydrogen fermentation, so you would also need to have some land to actually make the electricity. So think about, you need to have like either you need to have a windmill, or you need to have a solar power plant. So this takes up some space. You need to have a little bit of space for the bioreactor. Here they also factor in for direct air capture, so space for the CO2 capturing. But with this, you can make around 15 tons per year, and you can feed around 520 people. So this is a much more efficient way to make foods which with a really, really reduced land area to feed people, which means you could use the freed up land for other things. And you don't need too much land anymore to make foods, which is nice. But the disadvantage of the whole process is like hydrogen is like crazy flammable. So you probably all know when you think about hydrogen, you think about, oh, like explosions and it's flammable. And many people also, when we talked in the beginning about doing this, like many salespersons that we reached out to bioreactor, they were all like, oh no, you are crazy. This is like really dangerous. We can't sell you anything. And it's true, like hydrogen is like a flammable gas. And you have to be quite careful handling it, but it's something that's manageable. Like we also managed to handle natural gas safe in the lab and in many of our homes. And you can treat hydrogen and manage hydrogen similarly in a safe way. And one way of managing hydrogen is safely is to just not mix it with high oxygen concentrations. So hydrogen is only flammable if you mix it with more than 6% of oxygen. If you are below, so let's say you have a 95% hydrogen atmosphere mixed with 5% of oxygen, there is no risk of flammability. So that's one way of managing it. You just don't mix it in a ratio that would be flammable. Other ways would be you just make sure that there's no ignition source. And that's basically what we did to design our bioreactor. This is what you can see here. So that's our bioreactor, and here you can see a little time lapse of our biomanufacturing approach, where you can see in the beginning it's like a clear solution. So it's just the media, the mineral media with nitrogen, phosphates, and a few micronutrients. And then over time, it gets really turbid, which means the bacteria grows over time. And the setup includes, you see, it's like a glass vessel. So we chose to use a glass vessel so that there's no metal to metal contact, because this could result in some electrical discharge. And then potentially this can create a flammability risk. So that's one way we ensure that there's no flammability risk, is that we use a glass vessel, and the lid is Teflon, so there's also no source of ignition there. Different ways, or another ways we ensure that there's no flammability risk, is that we cultivate below the 6% oxygen, and we have two spargers. So we feed in hydrogen here, and then we feed oxygen and CO2 here, and then they only mix in the liquid media. And then in the liquid media, there's no source of ignition. So that's fine. And then there is a source of ignition in the headspace. There's a potential that there could be a oxygen environment that is higher than 6%. So that's why what we do, we have a nitrogen generator which generates pure nitrogen, and we flush the headspace with pure nitrogen, and then bring the whole mix of gases below the flammability limit before we vent it into the fume hood. So the whole thing sits in a fume hood, and the whole gas mix vents into the fume hood, and from there it's being diluted instantly. Another thing that we did, we don't use an electric motor, again, to prevent any sparks. We use an air-driven motor, and this is also an additional source of safety that we do. We also have like hydrogen sensors and so on, but this has been proven like really works really well. So far, no issues. We have to obviously follow really strict SOPs, but these kind of measures really ensure that we can safely cultivate with hydrogen in the lab. And we have like a 1 liter reactor, this is what you can see here, but we also have like a 10-liter reactor to really get enough biomass to then also do some fun food science experiments. So the process itself relies on hydrogen, oxygen, and CO2 gas, but to make protein, we need to feed nitrogen because the organism needs nitrogen to make protein. Otherwise, if we don't feed nitrogen, there's no chance of that the organism actually makes protein. And we tested like different nitrogen feeding protocols. So, for example, we feed only low feeding frequency. So meaning we only refeed at a really low frequency. We tested we refeed nitrogen at a high frequency. And we also tested like really adaptive feeding, where we constantly measure the nitrogen content in the media. And if it falls below 1 gram per liter, we refeed nitrogen. What you can see here is then the resulting nitrogen content. And this is what I meant in the beginning. So this process yields, especially when we adopted the adaptive nitrogen feeding, it yields a biomass that has like 83% of protein straight from the bioreactor. So we harvest the biomass, we wash it, and then we dry it, and then the powder has 83% of protein, which is like crazy. Imagine you have a process, you let it run for a few days, you make a powder out of it, and it just gives you straight 83% of protein without doing any refinement or anything else. So we were like really surprised, but also really happy about this, that this process actually yields so high protein content. And we tested a few other cultivation parameters. So for example, how much CO2 we feed, because it's also known that the carbon to nitrogen ratio highly impacts how much protein you get, but also in general your biomass concentration yields. And in our case, usually the carbon source is sugar, but we don't feed any sugars because it's like a completely from agriculture independent process. So our carbon source is CO2. So we tested different gas compositions where we fed in the gas composition 1, 5, and 10% CO2. And what you can see is that changing the CO2 composition, like first of all, highly affects how much protein you get, but also then your final biomass concentration yield. And this is just because the organism needs the CO2 to make these sugar components, which are then needed for the metabolism. So if we do not feed it enough CO2, it doesn't have enough carbon to actually make biomass. So it's kind of this balance that you have to strike between feeding enough CO2 and nitrogen at the same time to strike the balance between having enough material, but on the other hand, also have a high enough protein content. And what you get out of this, so here you can see the final product. So this is how the biomass looks like. So it's a little bit like where the picture, it looks a little bit brownish in the picture, but it's actually like brownish, reddish, because it also has some pigments. But the protein content under our like optimum gas composition and nitrogen feeding protocol, which yields high yield, so enough biomass, but also high protein. We get around 77% of protein, and the remaining is a little bit of ash, a little bit of moisture, a little bit of almost no lipids. This organism also makes PHB, which is kind of a bioplastic precursor, but it's basically only or almost only protein, which is really interesting for us because we are interested in finding new protein sources. But this is really the whole biomass. So imagine this is like whole cells, whole bacterial cells just dried into a powder. And from a food science perspective, this is something that might be not like crazy functional because these are like whole bacterial cells and they would just like precipitate easily in any food formulation and might not be really surface active. So you can't make an emulsion out of it or a foam or you can't extrude it. So it would be difficult to make food structures out of the whole cells. So that's why we were interested in can we establish a downstream process that would liberate the cells, so free the cells from the inside of the cell and disrupt the cells in the same during this process. So this involves usually like two separate steps. So you first have the biomass that you want to harvest, wash, and dry, and then you need to disrupt the cells and fractionate all the components from the cells. So this is what we do. We have our cultivation process. We then harvest the cells through a centrifugation process. Then you get this cell kind of paste out of it. So this is what you can see here. This is how it looks after centrifugation. We wash the cells because we want to get rid of all of the media components, especially the minerals and the ammonia, because the ammonia would kind of give us a wrong protein reading because we usually measure the nitrogen content and then from nitrogen content we calculate back to the protein content. So we want to make sure that all of the non-protein nitrogen is washed out. Then we dry it and then we did the proximate composition, so what I showed in the previous slide. And from there we then resuspend the cells. So technically you can also just go straight from the bioreactor to the cell disruption, but we wanted to have like really defined conditions. We redispense the cells, we disrupt the cells using a French press, which is kind of a high pressure homogenizer. We then get the disrupted cells, and we then separate it into the pellet, which mainly contains all the cell debris and the insoluble proteins, and the supernatant, which is like the soluble protein fraction. And then we're interested in how do these fractions behave in different types of food matrices. And here you can see a few results of the downstream process, how we designed it. So you can see this is how the cells and the cell suspension looks like after we get it from the bioreactor. And then here after centrifugation. And then we standardize the washing based on the conductivity. So we measure the conductivity over different washing steps. And you can see initially it's like really high conductivity because the cell suspension has so much minerals. And then you see we wash it and all of this gets washed out in the end until like the conductivity and also the nitrogen content is really, really low. And this way we established that after 3 washing steps, we remove approximately 99% of ammonium and the remaining salts. And this makes us confident that when we measure the protein content based on nitrogen, that we really, really only have the protein and we are not measuring these non-protein nitrogen components that we feed in during the biomanufacturing process. And then we want to disrupt the cells. So for this, we are measuring how much protein is solubilized, but also how many cells we have disrupted. So we look under the microscope, how many intact cells we have left, and we measure how much protein is solubilized. And this over different pressures in the high pressure homogenizer and over different number of passes. And when we measure this, we came to the conclusion that we need around 2 kilobars of pressure in the high pressure homogenizer to yield a optimum cell disruption efficiency, but also the maximum amount of solubilized protein. So this is what you can see here. One pass at 2 kilobar gives you like already a maximum amount of soluble proteins. If you rerun it through the homogenizer, there's not a lot of change. So really, you give the cells like one good push at a really high pressure, then you get enough protein out. And from there, you then separate it into your soluble and insoluble fraction. And the soluble fraction is really high in protein. So almost all of the protein goes into the soluble fraction. As you can see here, also the mass yield is 92%. And then this has a protein content of 82% without any precipitation. So think about usually when you do isolate a whey protein or a soy protein, to get to these high protein contents, usually you really need to precipitate the proteins or do membrane filtration to remove all other components. But here, naturally, it has a high protein content by simply fractionating without doing any further refinement. And when you look at protein composition, so here's just a SS page shell, the composition is quite different from what you would expect or what we know from our, let's say, the legacy proteins, because here we have a lot of enzymes and a lot of metabolic proteins. And these are not necessarily storage proteins. So all of these proteins that you identify and that make up this 82% of protein, they are like metabolic proteins, and they are, here you can see the hydrogenase, so the enzyme that is responsible for oxidizing the hydrogen. You have Rubisco from the Calvin cycle. You have the phosphorubilokinase, which is also a protein of the Calvin cycle. So you have these really metabolic proteins, which then also informs later on what you can actually do with these kind of with those ingredients in your foods, because this you obviously have to take into consideration. But the nice thing is that we can actually identify the proteins because all of them are described. So really high protein content, and much of the PHB, which is produced by this organism, ends up in the insoluble fraction. And then we did some preliminary like just functionality tests. So this is the soluble fraction, where we were interested in how does it behave, just a basic solubility profile. And you can see it behaves quite similar actually to what you would expect from most proteins. So really low solubility around pH 4 to 5, which is common for most protein sources that we use. So this is nice because this means maybe this soluble fraction can replace some other proteins with a similar solubility profile like soy or casein or whey, but really high solubility, so around 80% at pH 7. So this is like the good news from our like initial studies. So there's more to come. It behaves quite similar to a common food protein, even though it's really this high variety of different metabolic proteins that we found in our SS page shells. But one thing that I also wanted to mention is, and this is especially if you're like new to this field of protein analysis, biomanufacturing, and reporting protein content. Usually what is often reported on packaging or also in literature, it's based on the nitrogen content, but the nitrogen content varies depending on the protein composition, but also on non-protein nitrogen that is present in your sample. So nitrogen is also present, for example, in cell walls or in bases of nucleic acids and things like this. So nitrogen is not only present in protein, but it also shows up then in your nitrogen measurements if you use like the Dumas method or the Chiari method. And this has to be accounted for. So typically the standard is to take your nitrogen percentage and multiply it with 6.25, which works for some raw materials, but not for all of them. So this means if you use the 6.25 for a new raw material and you don't know exactly what the nitrogen to protein conversion factor is, you most likely either underreport your protein content or you overreport your protein content. So that's why it's important to establish your nitrogen to protein factor. And you can do this by just analyzing the amino acid composition. So technically, your true protein content is just the sum of your amino acids. And this is what we did here, for example. We just analyzed our amino acid composition. And from there, the sum gives you the true protein content, but it also allows you to calculate the nitrogen to protein factor. And you can see here, if we would use 6.25, which is kind of the standard factor, we would like greatly overreport the actual true protein content because the crude protein, which is like nitrogen times 6.25, gives you like 82% of protein. But if you use the actual factor of 5.2, then gives you the true protein content, which is around 60%, which is still a lot, but it's not 82. And that's why I think it's always to be like aware of this, that if you report protein times or nitrogen times 6.25, be aware that your actual value might be a little bit different. But it's still a lot of protein. So it's still 60% of protein without actually doing too much of refinement. And this is totally fine if you want to have this as a protein concentrate. It wouldn't qualify as an isolate, but it's already really nice for a protein concentrate. Yes, amino acid composition, there are a few highlights. And overall, I think the next steps for some of this would be to understand what is the actual, not only how much of the different amino acids are in there, but how available are there, so which would then inform the PDCAS value or the DIAS value and understand what is the actual protein quality. Yeah, I think this is like the key takeaway of the biomanufacturing. So we use hydrogen as the main energy source for this organism. We feed in CO2, oxygen, a few minerals, nitrogen, and then we get a protein ingredient out that is really high in protein without doing any agricultural work, without feeding any sugars. So we basically have the bioreactor, we plug it in, we plug the electrolyzer into the outlet, and then we can make foods, which is quite exciting. And this is like just a new way of making foods and food ingredients. And we are continuously working on this. So we're trying to understand better what are the functional properties of this based on solubility profiles and try to understand more how can we actually use it in foods, what kind of food textures can we make out of this, in what kind of matrices does it actually work, and just try to get a little bit better understanding. And now I noticed I already talked quite a long time, so I think I don't want to talk about the second part, but I think I want to skip this. I think if you're interested in a little bit of our extrusion work, I'm happy to chat about it in general. So if you have any questions relating to extrusion, you can just reach out to me, and then we can have like I think a general conversation. So I wanted to talk a little bit about our low temperature extrusion, but I think it would take a little bit too long. So I think overall, to conclude, I think it's really, really cool to be a food scientist, especially now with so much interested in finding new ingredients, especially finding new protein ingredients for different types of reasons. Because maybe it's more sustainable, maybe it's tastier, maybe it allows us to make less processed foods. Maybe it's like a really, really cool raw material and all these kinds of questions. So I think there's so much questions still to be answered in terms of using proteins for foods. But I really think we need these integrative approaches. So what I just showed you, like, I think it's not enough to only focus on the food biomanufacturing side, but we also need to understand once we have it, once we establish these new technologies, especially that allow us to make these new ingredients, we also need to understand how can we process them into an actual food ingredient? We need to understand how do they behave. So what are the properties that are relevant for foods? And then also how can we actually, what technologies can we use to process them into actual foods that then can be eventually consumed by people, because otherwise the impact of what we are doing is really, really low. So I think it's really important that we go like all the way from production to food engineering and making actual foods. And I think that's why food science is so cool, because we can do all of this, and it's so interdisciplinary that we can yeah tap in so many different areas, that is really interesting, and then hopefully make the food system a little bit better. Yeah, obviously none of this would be possible without the students and the postdocs that worked on all of this. So especially the hydrogen gas fermentation, my PhD student Carlos and our former master student Brana produced all of this data. And then for the extrusion, there's like especially our undergrad Nicole and prior postdoc Banu, who did all of this work that I didn't show. But if you're interested, there's like a really interesting publication on this. So you can just scan the QR code, and our research, the funding bodies that funded this, like the Foundation for Food and Agriculture Research and the Good Food Institute. And yeah, with this, I think I want to close. And yeah, I think I'm happy to take any questions that you may have. If you have questions regarding extrusion, I'm also happy to answer it. But if you have questions regarding like the biomanufacturing that we are doing, yeah, I'm happy to answer any questions.
Awesome. Great. Thanks, Lutz. That was really interesting. I guess I'll open it up to the audience. Does anybody have any I know I have a few questions, but I guess anybody in the audience, does anybody have any questions for Lutz? Okay, you can also put it in the chat if you have any. So I kind of had a couple questions. So, and I know you alluded to the fact that you're still doing a lot more on the characterization end now that you have like this really rich soluble protein. I was kind of curious, like, have you actually started trying to utilize it in any foods? And is there a particular group of foods that you see this potentially eventually being utilized in?
So we haven't tried really one specific food. We are planning to do some extrusion work on it, because we are really, really interested in extrusion, and we want to see whether we can make like the hydrogen meat alternative. So I think that would be really cool. But we haven't, as of now, we haven't tested it in any particular food.
In terms of one of the things that I like kind of jumped out at me is you sort of mentioned that when you have this protein fraction, the soluble portion has a very low amount of the sulfur containing amino acids. Do you think that would have potential like textural consequences because like you would have less disulfide bonds and stuff formed?
Yeah, exactly. So I mean, this is exactly the point where you then have to start thinking about what does this actually mean in terms of food structuring. If you have like maybe not so much cysteine, yeah, maybe you don't have a lot of disulfide bonds, and this could have impact on, yeah, maybe structuring interfaces or structuring a meat alternative, for example.
Well, have you thought about potentially like combining some of this, like so not solely using just this protein alone, but then potentially using it to supplement or sort of cut other sources of protein to add functionality?
Yes, I think that's probably what you will have to do anyway, especially in the beginning, um, where you mix this, for example, with a wheat protein or a soy protein to maybe overcome some of the shortcomings in terms of amino acid composition, but then maybe also in terms of how much you can actually produce of this and functionality wise. So blending is one thing that we maybe have to do, but we also are interested in, okay, does it actually work just as a standalone ingredient? And then if it doesn't work, then we probably will think about, okay, maybe how can we design kind of the mix to make it work?
Yeah, or it might be the other way. Like maybe it actually adds functionality to some limitations that soy and some of these other proteins have,
Yeah.
which I think could be really cool. It'd be
Yes.
really exciting. I probably shouldn't ask this, but have you actually tasted it? I know you're not supposed to for safety reasons, but you had
Yeah.
like you have?
So we haven't tasted it for, well, first of all, it comes from the lab. So
Yeah.
that's one thing. And the other is that the strain we are using is technically not CRAS approved yet.
Oh, okay.
So we think that the CRAS approval process of this strain is currently under consideration. We're not 100% sure, that's what we are guessing, because some startups also work with this strain. Um, but there's a strain that's already self-GRAS approved. It's not the strain we are working with, but another hydrogen oxidizing strain. This is like from a company that's based in Finland. They self-approved their strain for the US market, and they got food approval in Singapore, I think. So there are some of these hydrogen oxidizing strains that are already food approved. But the ones we are working with is not. And also, even if, because it comes from our lab, so technically we are not
Yeah.
allowed to taste it. I mean, I can't say what some of the students did. I'm not aware of it. But I don't think
Yeah.
that someone has already tasted it.
That's a good question, Kathy. So what's the protein's aroma? Just like you're not tasting it, but like there's just the smell because presumably you have a lot of it, right? So is there a smell that it has?
Yeah, I mean, we have a few 100 grams of it. Per batch, we can produce like 100 to 200 grams. So we have actually quite a bit of it. But the smell is quite neutral. It
Okay.
doesn't smell like a lot.
Okay, cool. That's a really good question. I guess another one, like, so you mentioned there's multiple different strains of these bacteria, but how easy is it to just like, so you have this one specific strain of the Cuprobacter or whatever it
Yeah.
is. How easy is it to go, oh, I want to actually just try this bacteria? Do you have to like re-optimize everything, or is it pretty interchangeable with your setup?
I guess well, I don't know because we haven't tested really different species.
Mhm.
Um, I guess it would be interchangeable, but probably to really optimize it, maybe you get like really low yields or low protein content. So I guess you can grow it, but maybe not in a way that would yield enough material or enough protein. So again, I guess as soon as you switch something, probably have to go through the whole optimization process again.
Okay. Okay. But like you could still use the setup though. It's just like you're optimizing like the CO2 and the nitrogen levels and stuff. Okay.
Exactly, yeah. I mean, that's what I hope at least.
Yeah. Yeah. Cool. Cool. I guess were there any other questions? It's really interesting stuff. Does anybody have any other questions for Lutz before we adjourn? Yeah, so I'd also like to thank you. So Kathy says thank you, and I'd also like to thank you, Lutz, for giving us this really interesting presentation about all the really cool work you're doing, and wish you the best of luck. Thank you,
Yeah.
everybody, for showing up, and have a great evening.
Yes. Thanks, everyone. And thanks again for the invitation. Thanks.
Yeah, thank you.
Thank you.
See ya.
Bye.
Bye, everybody.